Right. So hopefully everyone can see my screen. Maybe if someone in the chat can just let me know if they can't. Okay, so we just finished. We take a step back. We just finished fastq. I had some exercises, but we've moved rooms. So I'm just going to run through all the exercises we have at the end to save us some time now. But importantly, we learned what fastqs are, how we can read them in, and have a little look at their sequences and quality control. Most important thing to do with any fastq is to look at quality across cycles, across positions in the read. So from first base to the last base, do they change in quality, drop off, get better maybe at the very beginning? And also to assess the base pair content of your reads as you go across time. So perhaps we can just show you a little bit about what those last two things were, but I will run through the exercises in full. But importantly, this type of plot where we can see read quality dropping across time, time being cycles here because we start by sequencing the first base and we get to the last base. This is often to do with reagents running out. And then we may see some bias across time. You actually don't see anything here, but you may see bias across time also inside the base pair content. But more often you will see some bias often at the beginning or some bias at the end. Okay, so just keep an eye out for this, things which look different from your average fastq you're processing. So given that we have fastqs from genomics, often we don't really need to do much quality, just reviewing the data. Next thing we need to do is, let me turn on the video. Next thing we need to do is align our data. So this is actually our most popular course, most popular video. And I think it's because really this allows you to go from raw data you can get from any GEO, any published study, which is looking at high throughput sequencing, to something you can start to review in IGV. So in our previous sessions, we had a little look about how we can handle unaligned sequence data, as well as where we can retrieve genome sequence, full genome sequence reference files as Fasta format. In fact, we've shown you can get these from your BS genome packages. So typically, Fasta format look like this. We just have one line at the beginning of every contig, at the beginning of every chromosome, greater than, it's the name of the chromosome or contig, and then the actual sequence of that chromosome or contig. And eventually, you'll get to the end of the contig, and you'll have another line like this, which will say chromosome 2, and then you'll have all the sequence. FASTQ files are like a Fasta file, but with quality. So we have a header line, which is this, and that tells us where it is in the machine, what machine and where it was or is in the FASTQ sequencer, usually Illumina. We have the actual sequence. So like here, we actually have a sequence we want to align. And this is the sequence from our fragment. And then we have the quality for that sequence, and they're all paired here. So this is the quality for our first sequence. At the end, we have, say, C and a hash. So that's the quality for that particular base. How likely that base is actually truly that base. So, Illumina sequences aren't perfect. If we can't tell, it will give us a low quality score. So we're going to want to produce a file like this from our FASTQ file. And that's going to allow us, first off, just to visualize our data in IGV. We'll see very soon how we can turn a BAM into a bigwig, and we know how to handle bigwigs. Again, we want to look at them in IGV, but also this BAM file contains all the information we need for downstream processing, RNA-seq, peak calling, attack-seq analysis. We want something like this. So just to remind us, the SAM file has all the information from the read ID, the actual sequence of the read, the qualities of the read here, but additionally it has alignment information. So what chromosome it mapped to, what position, the position of its mate, and also how it's mapped. So here it's 28 matches in a row, 1 deletion versus the reference 72 matches. So SAM or BAM file will hold the information on actually how or where the alignment, where the mapping of this read is to our reference genome. So we're going to need a suitable aligner to do this. Place our unaligned reads, our fastq, onto the reference genome, the Fasta, to produce the SAM file. When I say SAM/BAM, remember SAM is the human readable version. It's just a text file with rules. The BAM is the binary alignment map, while SAM is a sequence alignment map. This isn't human readable, but it's what we want to produce if we want to do downstream analysis. I think that, you know, I can place aligners very broadly into 2 categories. We have our genomic aligners, which we will use for things like whole genome sequencing, ChIP-seq and ATAC-seq, and really anything where we expect a contiguous alignment of reads to the genome. Splice-aware aligners are a little different, and they deal with things like RNA-seq, ribo-seq, typically sequences coming from transcripts. Because transcripts have been spliced, they don't map directly to the genome, right? You'll have splice events, which are when exons are spliced out from the full-length exon-intron genomic sequence of a gene. And once exons have been spliced out, they'll be placed together into a transcript. So sequencing that means that we're going to have reads which span from one exon to another, missing out all the intronic genomic sequence. So we need aligners which are aware of where exons may be, either from us providing a map of where exons may be, gene models like a GTF, or perhaps they know the rules of splicing. And I think there's a lariat structure, which I can't quite remember what it is. I think it's AGUT, which is often spliced. So if you see this within a gene, it's often that we'll have a splice site there. So popular aligners for doing genomic or aligning to the genome include Bowtie, actually Bowtie 2 now, BWA, GMAP, and Subread. So I really like Subread, and we'll talk a bit more about it later. BWA has probably just been updated by Minimap. It's a very similar aligner written by the same person. But we're not going to cover BWA today. Popular splice-aware aligners, so things which are aware of this exon-to-exon junctions, this need to map reads across exon-exon junctions, is HiSat2, actually previously TopHat2. It's written by the same people. I think David, you might correct me if I'm wrong there, but written by, it's kind of a successor to that. SpliceMap, which is a little old now. We'll talk about it, but we're not going to use it. Subjunc, which is from the same family as Subread, it's actually in the same package. GMAP, we have GSNAP. And finally, we have the very popular STAR aligner. Okay, so we're not going to cover STAR today. If anyone's particularly interested, we can put together some slides on it. But we're going to cover, I think, Bowtie 2, Subread, HiSat2, and Subjunc. They all have slightly different features, but they will give similar, very, very similar alignments. Just the way they go about really both a combination of how they've implemented it, speed, memory efficiency, but also there are slightly different rules amongst these aligners, which can make subtle differences. So a splice-aware alignment, as I just said, splice-aware aligner is important for analysis of RNA-seq, where mRNA introns are spliced to stitch exons into contiguous sequence. And if we look at this IGV track, and this is over, I think that gene of interest we had actually in our IGV tutorials, where we see this kind of switch between these two exons being used, we can see the reads mapping from one exon to another here, these long kind of lines. This signifies that we have a junction. You know, a read starts here, and because it's been spliced, it actually ends up mapping there, there, and there, etc. So we have reads which map across exons. Very little reads which map within. We see some here, but if you look at the coverage, most of the signal is across exons, and then we have these reads which kind of span the two. Oh, gone backwards. So a few of the popular aligners are wrapped in our Bioconductor packages, allowing us to use that aligner from R, and I think most importantly, often they are closely tied, shall we say, to the Bioconductor objects we've already seen. I think growing to love might be a little strong yet, but we'll see. So Bowtie has R Bowtie, we have R Bowtie 2, because actually there is a Bowtie 1, and we have this package QuasR, which is a wrapper for a few different aligners. We have GSnap and GMap in GMapR. We have Splice Map again in QuasR. We have Subread and Subjunct, which are, I will say, the BRC favorites in R Subread. And we have HiSat2 in R HiSat2. But again, the author of R HiSat2 has written this up and implemented this in QuasR. Yeah, so QuasR has changed a little bit since we've started doing this course, only in the last 5 years. And one of the big changes is to now implement HiSat2 by way of this R HiSat2 package. So it makes it actually a really nice tool to do our alignment. So GMapper apparently isn't implemented on Windows, so we wouldn't want to use that. QuasR gives us this interface to Bowtie Splice Map, and much more recently, R HiSat2 or HiSat2, and it is available on Windows. So it's a nice package for us. We can use this in the class. R Bowtie 2 offers a wrapper to Bowtie 2 software, and was actually significant improvement over the Bowtie, which was packaged in QuasR. And then R Subread, actually very handily, is now just been updated to be included on Windows. Use this on Windows, and it is our recommendation for analysis. And anyone who really wants to do some reading, Subread, let's say, is in slight competition. I'm sure there's competition amongst the aligners with HiSat2 and Star, and in particular Star. So they've got a great paper on their explanation, which I really love this name. The R package, R Subread is easier, faster, cheaper, and better. Right? I think those first three are great for us, but the better bit is at least as good as some of these other aligners. Because my slight annoyance with Star, not annoyance, is unfair, is that Star uses a lot of memory. So I think it uses around 32 gigabytes for the human genome. You haven't got that on your laptop. R Subread, as you'll see, you can actually align it on your laptop, and it has modes to allow you to lower the memory, both for the alignment and for the indexing step. So big fan of R Subread, and I'll also point out this guy, Gordon K. Smythe, and also Wei Shi, I guess, but he's one of the founding members of Bioconductor, and he has written lots of other packages, including EdgeR and Lima, which means that this R Subread, they have paid very close attention to how it will integrate with Bioconductor objects. So great caliber, and also from the Bioconductor family, really. So we're going to again use some data, which is real data. We're going to take the data from our previous session, GM 12878, and we're going to use that for our alignment in today's session. To speed up the process, though, I've actually taken data from this sample for some of the illustration in the course. And what I've done is I've taken read mapping to actin beta gene, which is a really well expressed gene for this particular human genome build. And then even beyond that, I've taken just 10,000 reads, because there are 200,000 reads which map to that gene alone. Okay, so it's a very well expressed gene. So basically downsample this so that we can start to look, how do we align these 10,000 reads? We know where they're going to go. They should go to actin beta. That's where ENCODE put them. How are we going to do with our alignment? Using the tools we've already covered or covered today. So latest paper, is that the same one I just pointed to? Yes. Because our subreaders also now got modes for single cell, so they allow you to go straight to the single cell counts as well. So they really are updated frequently. Like all Bioconductor packages, we can just Google the thing, and then copy the line here to go install that. And I've got this line kind of written out here, but once it's installed, you do library, our subread, and we're ready to go. We're also going to need some reference data, and as I've said before, you can use your own Fasta files for this. So if you have a Fasta or a genome reference which your lab is liked or used, you have previous papers using that particular alignment, you're not going to change your lab's policy just for your analysis. So you can use your own Fasta files for this, but we're going to start by taking advantage of some of the Bioconductor annotation, which is great because it's version controlled. And we're going to take BS Genome, H Sapiens, UCSC, H38. So once I've installed this, I can just load the library, and we know that once I've loaded the library, there's an object available to me, BS Genome, H Sapiens, UCSC, H38 in the environment. So as soon as I load this library, that object is within R for us. So for build an index, we need our reference sequence in Fasta format. So a lot of programs, even if they're implemented in R, they still want you to write the object, the sequence to a Fasta. I think later on in the course, we're going to show you how you do this for a full genome. But just to save ourselves a bit of time today, I'm just going to use our standard indexing on our BS genome object. I will take out chromosome 7. I will set that to a DNA string object, and then need to make that into a DNA string set object to write it out. So the easiest way to do that is to create a list with a named list with one element called chromosome 7 here, a string I just extracted from our object here. Once I have that, I can just turn that into a DNA string set using the DNA string set function. Yeah. So then I have my DNA string set just chromosome 7 at the moment, and I can write that to a Fasta using write x string set, give it the name of the DNA string set object, and tell it to write it to a Fasta, QuasR7.far. Okay, it would be nice actually if you could just write out the BS genome straight to a Fasta. We can't, but I can show you how we can loop through the chromosomes to write out the full Fasta file for our RNA-seq, ChIP-seq, and ATAC-seq. We'll do the full thing now. So right now we have the chromosome file from the BS genome object. So we have a Fasta file. And the next thing we need to do is try and build an index of our reference genome. Okay, so we're not going to use the Fasta directly. We're actually going to build an index. I'm going to try and make it easier for the aligner to do its work by first building an index from this Fasta file. Note, we're going to build an index multiple times for different aligners. So every aligner, or most aligners, use a different type of index. But for each aligner and each species, you just need to build an index once. So I'm using our subread and I'm using HD38. I just need to build an index once for that Fasta file, and then I can reuse that index over and over again. So this can be quite memory intensive. It can take a bit of time. But once you've done it, you don't need to do it every time you do it. So by default, the memory used when indexing, building an index is limited to 8 gigabytes. This is often quite large for a laptop. I don't know what laptop you have. I mean, nowadays, maybe not. But if you're doing other things on your laptop at the same time, 8 gigabytes free might be a little much. So we can actually control this when we do the index building, and we can do build index. We just need to tell it the name of our index. Here, I'll just call it QuasR7. The file to use for our index, and then the total number of memory in megabytes that we're allowing the program to use. So if you lower that to 1,000, you'll notice that your index is going to build slower, but it's not going to swamp your memory so that your computer can't do anything. Okay. Sometimes if you're on a cluster or something, you're limited and it's going to kill your job if you go over that amount of memory, you do need to control that. I just want to take a second to talk a little bit about what an index is. And I'm going to do a real toy example I was building in our break. Okay, let me make this a bit bigger. Okay. So all of these indexing tools have a very similar trick. And effectively what they're doing is building an index akin to what you would get in a book. Okay, so I'm going to show the process, very simple version of indexing here. So given our genome of, I don't know how long that is. I don't think I've fully indexed this. Okay, I could have read it from here, right? 20 base pairs long. We're going to then index that. And what we would do is starting from our first here, if I'm using an index with my KMA set to 3, I'm going to start off by looking at the first 3, and I will make a mark in my index, and I will say I have ACT at position 1. I will then work through this, CTG position 2, TGC position 3. This is what the computer will be doing when it's building an index. And every so often, it will come across something it has already come across, right? So ACT, somewhere here, was actually the start. So I'm going to put it as 9 because it's here at the 9th position, but I'll put a star just to let us know that, you know, it's also at another position. And we can work across the full, in sliding window of 3 till eventually we get the ATA, and then we'll get the TAT at the end. Okay, so we have a position of every 3ma, and then we can collapse this so that we don't have duplicate 3mas here. And I will say, okay, ACT is at position 1 and 9. Okay, I might have missed some, so I was just doing this by hand as we in the gap. But we have a TCA. Actually, I could use that to kind of highlight if I've missed anything. But we have a TCA here, which was at 6 and 14, and I'll label it as 6 and 14. Okay. So as you move through a whole genome, usually in 8mas, but you will find that you have lots and lots of repeats of an 8ma sequence. And so in a way, we're actually compressing the genome quite a lot by storing it with this information. Okay, we don't have the KMAs multiple times represented, they're just represented once. We can say the start position of this 3ma. So with that index now, the first step of alignment is actually very simple. And it's the step after using the index that the aligners start to diverge, right? It's there that once they've done this simple problem, that they will start to have different algorithms to do the next bit fast. But given we have a read of ACT CGT, we can then split that into 2 subreads here of length 3, so the same length as that of my index. I'll have a subread of ACT and a subread of CGT. With these subreads, then I can quickly scan my index and I can say, okay, ACT I got here, and it's got a position of 1 or 9. If I go down to CGT, we can quickly scan through, and then we have CGT at 12. Okay, so now I have 2 potential positions for my first subread, 1 for my second. I could be at 1 or 9 for this one. I could be at 12 for this one. And then if we actually think about this logically, and this is what a computer would do, only 9, if I add 3 to 9, right, it's going to be 12. So the only position this would really have worked at was if ACT was at the 9th position, which would allow these bases, 3 base pairs further along, to be here at the 12th position. Okay. So very simply, with like a read 6 base pairs and this tiny genome, we would be able to make a decision from this where this read should map to. Okay, so that's the very simple basics of how an index system works. By doing this up front, then when it goes to align, it's going to save it so much time rather than having to scan the full genome over and over again, right? Or hold the whole genome in memory for when it's trying to find out all the potential positions for ACT and then, the next part of it. Obviously, it gets more complex if there's mismatches, and this is where some of the algorithms inside these tools start to compete with each other, best ways to deal with that. So building that index is essential. It takes a bit of time, about half an hour. But once you've done it once, you don't need to do it again. So you can just run that and then align. Now we have our genome, we can align our fastq data to it. We can keep our fastq data spliced. Yes. So you get to a yes. So splice aligners have slightly different rules in that they know we've told it up front what the potential splicing could be. So it re-implements that inside its indexing. You also have splice aligners that they will try, if you can imagine, reads are often 100 base pairs long. A lot of the read and these little fragments we just looked at, sorry, it's on the R, they will map within an exon, and then the strategy for the aligner is saying, okay, I have two parts of the read, these two subreads map here, these two subreads map in a different exon, but I know that there's an exon between them. Can I now fill in the rest by usually some type of local alignment? So this strategy of aligning to the index is really the first pass. And then given how they're arranged, given if it's a splice-aware aligner, is there a splicing between them? It will then try and fill in the remaining reads or remaining sub-reads of that read. Great, so then we're going to do align. And remember, this is a standard alignment here. This isn't spliced aware. We're not using the right function yet. Sorry, I got another chat there. Great, thank you. Thank you. So here we'll do align. We'll give it the name of the index built, so the same name as here. We're wanting to align. So here, data sampled actin. I tell it the output format's BAM. I actually don't know what the alternative is from subread, but then I'll tell it the output file, and it's then going to write that to our subread, no splicing, sampled actin.bam. Okay, so now we have our aligned data as a BAM file. We got to do two final operations on our BAM file to make it ready for use with external files. Okay, so I'm going to show you how to do this. It might be worth noting that different aligners will output your BAM in slightly different orders. So subread likes to output your BAM in read order. So the read name is used for the ordering. Whereas some tools will actually sort it by coordinate for you. So it's worth checking, and we can check by looking at the SAM header whether or not our files are already, our BAM files which are generated are already sorted. Sorting is, again, quite a computationally expensive task. So to do sorting, we're actually going to use a really popular piece of software called Rsamtools, but we will use it by way of R through the Rsamtools package. So Rsamtools, if I just search this, I want the GitHub, there we go. So Rsamtools is written in C, so very, very low-level language, and it's very popular. It got updated to use a slightly different library set, HTSlib, quite recently, which is again faster, but lots of programs, lots of tools take advantage of this piece of software. And they do it really just by compiling all the C+ libraries into whatever language they want to call them from. So you have a Python equivalent called PySam, and we have R. PySam's really nice as well, actually. But we have the R equivalent in Rsamtools. Like with any type of Bioconductor software, we can install Rsamtools using the Bioconductor friendly way, where we just Google it and we copy and paste this bit into R and then press enter, and it will go and get the install that package, but also all its dependencies. Okay, so once that's installed, we can just load it with library Rsamtools. Okay, so one of the first functions which is super useful is the sort BAM function. So the sort BAM function takes 2 arguments. It takes the path to the BAM you want to sort, and slightly strangely, it wants the prefix for the sorted BAM file output. I'm sure there's a reason, I just haven't come across it yet, but that means you don't put .bam here. It's not asking for what the BAM file should be called once it's sorted. And once you run this function, it will run this and it will create, if I put this as my prefix, a file called sorted_actinb.bam. But again, if you put the .bam here, you will end up with sorted_actinb.bam.bam. It's a bit annoying, worth noting. Okay, we can also then index that BAM file. Okay, so remember we said with every BAM there was an associated bam.by. That's not always the format. Sometimes it's just .by. But most programs will look for either version of this in the same directory as this when they try and load your BAM file. They will look for a .bam.by or a .by. This is what IGV does, for instance. It just checks to see if you've got that file. By pressing index BAM on this file, we will generate the indexed file, and that means then we can do random access, and we can start to import it into programs like IGV. If we want to get a very quick overview of the alignment, we can actually use this function quick BAM flag summary on our BAM file, and it gives us an output which isn't super friendly for actually capturing it and writing out to our table, but it does print it to the screen, so we can have a quick look there about how well we've done with the alignment. So we'll do quick BAM flag summary, and it will go through, and actually it checks, I'm not sure if we can remember, but we have various flags within this file, including our bit flag here, telling us the type of alignment. Is it a unique alignment? Is it first in a pair, etc.? So this flag tool is going to go through and just quickly try and build up statistics on our alignment. What we can see, though, is that we don't manage to map all the reads which ENCODE has managed to map to this gene. Right? We actually have 97%, I think, mapping rate here, which is pretty good. But we're definitely missing some reads. So it's nice to go and actually look to see what those reads are that we're missing. First thing we can do is go and look in IGV at our BAM file. And we can see we have coverage really piling up along the exons. Some here, like between the exon, but obviously not much compared to the coverage here. But we are missing something, right? And that is we do not have any splice junctions. So even though we've managed to align a lot of our sequence to this actin beta gene, actually we don't have any of the splice junctions which we want to kind of map from one exon to another. So I'll say at this point, the authors of our subread will say that's not a problem. They say you get enough alignment, 2 exons using just subread, and you don't really benefit from bothering to map the splice junctions. I believe them in terms of gene expression analysis, but I still want to see my splice junctions, and I'm willing to wait an extra 15 minutes to go get them. Okay. So if we're missing these reads and we want the splice junctions, and honestly, you can read the paper on their comments on whether you need them, but I'm going to go get them because it just takes a little bit more time. So to run subread in a splice-aware manner, we have to use not a line like we did here. We'll use a new function called subjunct. So subread, but for junctions. We actually, by default, don't need to provide any other information. So we don't need to provide a GTF for this. It will guess where splicing junctions should be based on the known lariat structure that causes exons to be spliced. We just need to give it the same index, the path to the fastq, tell it the output file, and now I'm going to tell it to write it to this file instead. We have to do the same things every time we do this. We sort our BAM file, and then we index it so that we can then use it for IGV and other programs tools. Okay. So now, if we have a look at the alignment, we can see we got marginally better. We actually managed to capture a few more reads, but not that many more reads, really. So I think we went from 10 or 20 more reads here. So really not that many more reads. But then if we actually go look at it, we can go look at the junctions and we can start to see, yes, we have now got reads spanning these junctions. Okay. So an interesting point here is that the reason we didn't get a lot more reads mapping is because subread has already mapped them in this strategy. And it didn't map them by mapping them across exons. It actually did it by clipping the ends of reads. So it said, this read maps, this read maps best to this region apart from the end of the read. If I remove the end of the read, does it map better anywhere else, or is this still the best region? And that would be the post-index kind of alignment step. Once it's built enough subread that it's saying it's most likely this region, it will try and effectively clip off the end of the reads to make sure it can align to an exon. Okay. So actually, when we run this in a spliced mode, what we're really doing is allowing the reads which were previously clipped, because we weren't looking at a splicing alignment, and now they're being actually mapped across exons. Okay, so it's just changed the way that those reads map. And that's why we're not seeing suddenly a ton of more reads. But then still, what is happening to the missing 3% of reads? Actually, if we go into, we take the encode data directly, we can start to see really where these reads have gone. And actually, a lot of these reads are mapping in very improbable ways. So in this case here, we can see that these reads here, which map to this gene, are actually mapping from one exon quite some way, and they're actually mapping over the next gene and to a gene which is 3 or 4 genes down. Okay, so this is really a feature of STAR versus many of the other aligners. STAR is really going to try and align these reads even in slightly improbable ways, which, depending on really what you want, can be up to you. What I will say is that when we come to actually counting the reads aligning to genes, so when we want to do something useful with these alignments, any reads which map to more than one gene, we're just going to throw away. It's a hard, fast default rule, even when we're using STAR alignment. So these reads which start in actin and then map miles away, they're not going to get counted anyway for when we do the actual differential expression analysis. They're often not counted for things like transcript abundance analysis. So in this case, this 3% of reads we've lost are in fact not useful to us. So I think that's kind of an interesting feature of STAR versus some of these other aligners. It is often aligning into slightly improbable ways. So I like subread, and I would recommend you should use this for most of your analysis. Bowtie 2 is a very popular package as well, and is actually used in some packages which don't necessarily do standard alignment things. So things like ClipSeq, where we're aligning mRNA reads, which are very short. Bowtie can be quite popular for that. So we're going to first off just install it on BioC Manager -- install Bowtie, and then we can load the Bowtie 2 library. Just like with our subread, we need to build an index the first time we run this Bowtie aligner on a particular genome, but we can use the same Fasta file we generated earlier. I will just now run Bowtie 2 build, give it the path to my reference Fasta, and then I give it the path I want my index to be built out. One slightly annoying thing about Bowtie 2, or our Bowtie 2, is it was really built for working on the command line. So it doesn't like to deal with zipped files itself. It was expecting you to do something fancy at the command line where you unzip them while piping them into the tool. So that just means that we have to unzip our fastq files ourselves. So we can use the gunzip function. If you don't know how to unzip these GZ files, we're going to use gunzip from inside R. But to actually use that, we need to load this r.utils package. I believe that's actually installed when you load R, when you install R. It's just that we don't load it by default. So load library r.utils, and then we can gunzip, unzip our fastq file. Once we've unzipped it, we can then pass that unzipped file to the bowtie2 aligner. Bowtie2 takes the chromosome, the index name, the sequence, and then it's going to take the output to the argument SAM output. So again, just like we had this fastq.guz, we needed to unzip it. Bowtie2 is slightly painful in that it actually doesn't produce a BAM file, it produces a .sam file, which is annoying. We have to do the translation to a BAM file. What's most annoying about our Bowtie2 is that by unzipping the fastq and by making a SAM file, potentially you're creating two very big files here. Indexing it, making a BAM is much smaller than a SAM, and a fastq, which is zipped, is much smaller than a fastq. So maybe just transiently, you will need to create these two big files. Perhaps you would delete this once you've unzipped it so you can just keep the zipped version. And for the SAM file, we're going to have to use this function as a BAM, again from the Rsamtools library, which is just going to translate our SAM file to a BAM file called .bam, whatever this name is. And then it returns the name of the file actually to this, from the output from the function to this object here. Again, just to be sure, we're going to then sort our BAM file. Again, not providing the index. Not providing the .bam suffix. And then we'll do index BAM on the sorted file to finally get a BAM file, which is bowtie aligned, and we can start to work with. So aligners have a variety of cost benefits. Our bowtie, it really is a significant benefit has a significant benefit compared to bowtie, which is slow, memory heavy. So our bowtie 2 we would typically use for things like ATAC-seq, ChIP-seq, whole genome sequencing, and you just see a lot of bowtie 2 in written into programs by other people. It's a very popular aligner, and it's kind of a little older now, so it's well known. Our bowtie 2 you can't use for RNA-seq or riboseq, right? And Splice unaware aligner. So we can use it for lots of our things, but not really RNA-seq or riboseq. Some of these wrappers we've been looking at have multiple aligners built into them, and one which is really good is QuasR, because it seems to have at least bowtie splice map, but most excitingly for us, it has this wrapper for HiSat2. Okay, and actually, not sure if we can bother looking this up, but our HiSat2 is very simple in the way that it wraps HiSat. But it's this author, Charlotte Sonesson here, who kind of works both in the QuasR package and in the HiSat2 package, who kind of helped it all wrap up. And then she joined as an author for the QuasR package. So QuasR is updated quite a lot since we started doing this course, and I think one of the big things it added was HiSat2. So again, like every other package, we need to go Google it or install it using this. And once we've installed it, we can do library QuasR, and we'll have all the functions available to us. So one of the main functions in QuasR is the QAlign function. It's a little different from the other tools we're using in that it actually tries to do a little bit more of the work in R. So we're not going to just provide a fastq file We actually provide a tab delimited file containing fastq locations and sample names. And for a reference genome, we can actually provide a Fasta file location, or usefully, we can actually give it a BS genome object. So this is the first aligner, which is really properly integrated with the Bioconductor objects. We can use the BS genome objects like BS genome, H. sapiens, HG38, UCSC, which contains the full sequence for the genomes we want to align to. So, but importantly, you can also give it a Fasta file. So you're not stuck just using the BS genome packages, because that would be just as bad as if you're not able to use them. So first thing we need to do is make this sample table, and we'll do that very easily. We'll just make a data frame, and we'll have file name in the first column, and that tells us the path from where we are to the file, and then the sample name, we'll give it sampled active. Make that into a data frame, and then we can write that table to a file tab separated, no quotes, no row names, and then this is roughly what it looks like, or is what it looks like. Once we have done that, we can then just do library QuasR, and then for the QAlign function, we just give it our sample table, and we tell it the name of the BS genome object we want to use. Okay, so we don't need the BS genome object loaded. You give it the name, and then it's going to stitch that together, and then load the library itself, get the object, extract the Fasta, write that to a temporary file, and do the alignment. Yep, so that's exactly what I just said. Internally, QuasR will create a Fasta file from our BS genome prior to alignment. We can also provide that Fasta file, which is obviously very important, and we can align our samples to this. Importantly, as you may have noticed, there isn't an index step here, right? It's just doing the alignment straight away. It does have an index step, it's just hidden it from the user. So whether or not I do an alignment with this particular genome, it takes a hash, it takes a way of recognizing that we are using this particular object or this Fasta file. It will create the index the first time it runs it, but the second time it's clever enough to go, okay, I've already created an index for this particular file. I can just carry on with the alignment. Okay, so it's just doing that little bit of work for us under the hood. I don't recommend using QuasR in this mode though. And usually we had QuasR in this class because it was the only one which actually worked on Windows back a few years ago. But now with the RHiSat2 and that being available for Windows, Linux, and Mac, actually QAlign suddenly became that much better. And for aligning to splice junctions in a spliced way, RHiSat2 is a really good alignment to do that. So to take advantage of this, we can either go and, remember there is a RHiSat2 package. It has its own manual. I'm pretty sure here they say, use the QuasR package somewhere though. As at least Charlotte Sonerson for sure says that. She recommends to use it through the QAlign package. So to do that, we just do the same inputs, the same sample table, the same Fasta. We just need to say aligner RHiSat2. And rather than using the Bowtie 2 or the Splice map one, now it's going to use this aligner. Remembering though, because we're using a new aligner, it's going to make a new index. So if you did run it like this, and then you ran it like this, it's still going to have to build a new index because we're running a different aligner under the hood. Okay, so that's the end of the session for today. I think we did that with roughly in time. We have some exercises on this. I will run through this. I have to go to a barbecue for the Fuchs Lab at 5. So I'm going to run through this a little earlier at 4:30. I will make a recording and put it on YouTube. The alignment might actually take some time as well, because you're going to build a index, you're going to wait for it to align, etc. That's quite cool. We're going to align the fastq for that IGV we did in the IGV session. But I will come back at 4:30 and run through it anyway. If any of you are still going, you can watch the YouTube video. That will have the solution at the end of it. Just catch up there. Anyway, so thank you all for today. I'm sorry I had to change room. I know some of you were in the actual room with me. I probably caused some confusion on Zoom. We had some issues with the projector today, hopefully resolved for our next session. Okay, I'll speak to everyone soon. I will be back at 4:30 to run through these examples, but I will record it if you guys just want to hang on and wait for the recording and give yourself enough time to read through. No problem, no problem. I'll speak to everyone soon. I'm going to stop the recording for now, and I'm going to stop sharing screen. I'll be back at 4:30, but everything will go on YouTube by the end of the day. Right, so I was just going to run through some of the exercises we were covering today. So what do we do in the first one? Plot the occurrence of A, C, T, A, G, T, C, and N in the read with ID, this ID using ggplot. So we're going to have to do a few things here. We're going to have to first read in, find the read with this ID, and then plot the occurrence. So we'll load the short read library. We'll set our file name to be the heart body map fastq as described here. And now that we've loaded the library, we can use read fastq, give it the file name, and we should just be able to then read this in to our short read q object. Next thing we want to do is extract the IDs from our short read q object. I'm going to turn that into a character just so you can match it with this ID I provided. Okay, so we'll use that match. It'll give us a true false. The subset our fastq just down to our short read q object, just down to the fastq entry we're interested in. Then perform sread on this short read q object, filtered down to this particular read. And on that, we can do the alphabet frequency and just extract the values for the bases we're interested in, A, C, G, T, and N. From that point, it's very similar to what we've done for Fasta plot. So we just load the ggplot library. We make a data frame because ggplot wants data frames containing the names of the bases. So these names, and then the actual frequencies we've extracted here. And then we pass that to ggplot, give it the bases, the frequency, and the fill. So here, X will be the bases, Y will be the frequency, and the fill will also be the bases. Geom bar, but stat equals identity, because we don't need to calculate any stats here. We're just going to take the actual values of AC, G, T, and N and plot them, and we'll do theme BW. Okay, so no ends, all GC rich. Create a box plot of quality scores for the first 1,000 reads using base graphics. Just base graphics, a little easier here. So we will extract the qualities for the first 1,000 reads using the quality function. And then we can use this trick we saw in class where we'll turn the qualities into a matrix. So as all quals matrix, I will then assign some column names to our matrix, and I just want to basically add cycle just to the number of the actual cycle. And then I will do box plot on that, and box plot can take a matrix. So it's going to create for every column a box, and we can see the scores dropping out over time. Plot the ACGTN bases over cycles using ggplot. Well, we'll use the alphabet by cycle on the result of sread call on the short read q object. So this will give the sequences, and then we can pass that to alphabet by cycle to give us the frequency of bases per cycle. We can then extract from this matrix just the rows to do with the bases we're interested in. And then I will create a data frame from that by giving it the base, the frequency, and I'll take the frequencies from the A column here for A. And I have to add in the cycle myself, but I know I had 75 cycles here. So once I've done that, I can then bind all my frames for the different bases together, one big frame, and I can pass that to ggplot now. X being the cycle, so along here is the cycle, Y being the frequency of the base, and then the color of the line being the base. Geom line being DW. For anyone who was looking at this and thinking, well, why do we drop down here? Because actually this data had a mix of reads in it. I think it had 50 base pair reads and it had 75 base pair reads. So this dip we see was just the fact that there were no more reads, or there were less reads which were 50, and then we were just to the 75 base pair reads. Okay. We can also see, like at the beginning, we have this quite a high number of ends in the first two bases. So that might be something to filter. How many reads are in the original fastq? Just get the length of the fastq. Sample 10,000 reads. So we'll use fastq sampler, and we'll just set the n to be 10,000. Give it the fastq. And once we've built the sampler object, we can just do yield on it, and it will produce our fastq object of 10,000 reads here. Okay. Count the number of duplicated sequences. So we can do that with SR duplicated. It will mark, or give us back a true false list of whether or not a read was duplicated. And then we can just run table on that true false list. That tells me I have 6,000 duplicates, 3,913 non-duplicates. If we want to actually get rid of the duplicates, we can just do myFastQ 1000, so the short read Q object. And I just give it the logical of all those which were duplicates, and then this will filter down just to the duplicate sequence. So actually, if we want non-duplicates, we should put an exclamation mark there to reverse the logic. Trim the resulting non-duplicate reads to remove reads with a succession of 10 bases falling below the quality of 5. So we saw this in class. We just pass in our short read Q object. Here, the one where we've actually kept duplicates. And then I want 10 bases in a row, which will drop below 5. And then I can just use my trim tails to actually run that. Okay, now plot a histogram of the resulting read lengths using base graphics. And we'll just use the width function to give us the length of the reads which are left. And then I can just pass that to the histogram plotting function. So I'm going to give it the widths of all my reads, pass that to the histogram function. And you can see we have really a distribution of 50s and 75s, which is probably a reflection of what we saw here, 50s and 75s. Filter out reads less than 32 bases and write to file. So we'll just do our trimmed non-dupes here. We'll just take those with the width, which is greater than this cutoff. So we'll filter it down using standard indexing here, and then just write that out to a fastq file. Finally, just to tidy stuff up, I'm doing unlink. I delete the fastq. That's just because fastq files tend to be big. In this case, I want to delete it. We can delete stuff in R just by using unlink, pointing to the name of the file. Great, so that's the end of the session for today. I will see you all on Friday.